So the last presentation of the day will be covered by Mandy Chen, and she will be presenting detecting DNA-based modifications on nanopore sequencing data.
So, hello everyone, thanks for staying to the end of today's talk. And I'm going to talk about some analysis we did earlier on nanopore sequencing data. So, continue with Pepe's talk earlier. We are working on this kind of new data called nanopore sequencing data. And in our case, we're using the data generated by Oxford nanopore sequencing. And so it basically works in this way that you have a pore on the membrane. Right, so nanopore sequencing, the biological sequence is going through this nanopore on the membrane. And then you have detectors, which is sending signals and receiving signals. So therefore, you can record the current. There's a current change in each base going through this membrane, and so there is a different current disruption that it can be monitored on the biological sequence. So and the modifications are also will have impact on this current, and this is the ground that we're on to detect modification in DNA sequence using nanopore sequencing data. So what about the data actually looks like? I don't have an example. So it's basically when each base is called going through these monitors, it will generate a set of signals. We call it raw signals in this case, and they are basically a set of integers. And these integers are vary. It depends on the bases going through the membrane. And the motivations on this project is there are some existing methods when we started in 2019. There are two types of different methods. One is based on the models based on base calling errors. By base calling errors, I mean that when you do the base calling, and you will obtain a set of features. In this case, these are the base quality, the average base mismatches, the insertions and deletions for each DNA nucleotide in this case. And then that was done by Epinano. It's a lab based in Barcelona, I think, and they use a PCA model to do the prediction on methylation states. And the other set of methods, particularly these two methods, are using neural networks, and particularly using recurrence neural networks. And the input feature in this case is the features that extracted from the raw signals. And the raw signals, as I just said, is a set of integer numbers. And then, so the input in their models is the signal mean and the standard deviation and number of signals, these kind of statistics. And also the input, the base called sequence of the DNA, which is the nucleotide sequence. So based on that, these are basically just these two types of models I just described. And so we think of if we can combine these two sets of information, will the prediction be improved? And that's basically what we try to do in this project. So the framework we built is showing this cartoon. So we built two sets of modules. One is the base calling error features. One is the signal sequence features. So we're kind of combining these two methods, these two types of methods, and the features they're using. And we hope for that we will get a better accuracy than any of them. and then this is the pipeline we're going through for the methylation calling. So first of all, you get the reads, and then we extract these two set of features, and then we input into our neural nets architecture, and then we'll do the read predictions for we'll do the read predictions for each of the we'll do the read predictions, and then we'll combine those read predictions for each of the position to get a position predictions. So basically, we're doing two things. One is the read-based calling on modification, and the other one is the position calling for modification, which should be more interesting in a biological wise. And then on that side is our architecture of the neural nets. So instead of recurrence neural nets, we use convolutional neural nets because we think that's more efficient, and we suppose that it will better capture the natures on this type of data. So, any questions here?
I have one question. So when you say you have mean currents, I guess that's within a window, is it?
Yeah. So
is there an issue in terms of defining it for the window itself?
Right. Yeah. I should explain this. So when you have a long sequence, we don't use the whole sequence to predict each position. So we have a window that's slicing from the beginning to the end, and it's a set length window. And we are sliding this window, and we input this the window size sequence into the model, and we try to predict the middle base in this.
So basically, you have flanking positions as well included in the model.
Yeah.
Okay. Right. But then, is it always so? Can you basically move this thing through with the constant speed, or is there actually a variation in the speed such that the size of the window does not always correspond to the same number of bases?
Actually, in our case, it's not an online method. So you already, when you process the long sequence, you already chop it into window sizes.
No, but what I mean, like within a window, let's say you have flanking region.
Right.
Do you always cover the same number of bases in that window, or could it be that if something goes, if I mention the string going through the nanopore, is it sometimes moving faster, and so you get the same window but more bases or less?
But I'm not sure I understand the question, but you're talking about. Do you want to answer that?
I can answer that. So basically, it is supposed to be moving at constant speed, and the thing is that the readout is not only one. So for one nucleotide, you don't have one readout of the core. You have several readouts of the core, right? And you're taking the mean over that. But the thing is that we have some pre-processing steps where you have like this set of currents that are assigned to that current. So that's already set. You will always take a window of 17 in DeepMP, and that set of window will never change unless you change it in the method itself.
Okay. Right.
My question is kind of, imagine you have a given window, somebody gives you a signal, and then now I ask, is there 7 or 8 base pairs within this window?
But that is done prior to it. So that's
not what you're looking at, basically.
So you have already the currents assigned to every nucleotide.
No.
So every nucleotide will have assigned some certain currents by the base caller and by further methods that you can use before entering DeepMP.
Yeah.
So the method is not about that.
We are not. So the currents are set of, like, for every base you have a set of, you have you have a set of integers. So you might have different length of integers for each base. But this is not what we input into this model. We need to extract the features we use, which are these kind of, the mean and the median of this raw signal. And then we input that, we construct that into a fixed length window. So that will be, in our case, we're using 17 bases. So we'll construct all these features for each 17 bases and put it together and input into the model. Okay.
So the width of the window is not in terms of the signal or the current, but in terms of the already inferred bases.
Yeah.
Yes.
Like one nucleotide can have one current, and then the next one have 150 integers of currents, like values of currents.
Yep.
So what is the input size?
You mean the window length, or? You mean the dimension? So it's showing here. Right? We have the sequence. We call it K-mer, which is the nucleotide sequence. We embed it with 1 hot, so that will become, I think that was 5. So they become 5, and then there's 4. You add it up, there's 9, and the other signal sequence features also the 1 hot embedded sequence with all these 4. So this is L times 5, and L is the window size. L times 9, and that's the L is the window size. Okay.
How about instead of taking these discrete values, feeding the continuous value?
Actually, because the current we have are integers. But when you compute the mean, and it's not necessary to be integers, right? But the raw signals are integers.
Well, can't change that a lot.
Then that is the question for the sequencers and the people who do those experiments. Not our. It's not. Yeah.
It's super noisy though. It's really noisy. Like for one nucleotide, like if you take the whole spectrum, like you have really a lot of jumps. So, and the previous methods had used that, and we thought that it would be interesting to use those features.
I think we have a pipeline doing that, which we didn't build that.
Yeah.
So you can check out the details on the paper.
Yeah, so because I have a feeling for neural network, if the dimension of the number of features in the input is small, for example, from my experience, classical machine learning algorithms perform better than neural networks. But if you have a complex input, for sure neural networks would be. So maybe if you give this signal, maybe the network can, learn to avoid the noise and maybe learn some useful features.
That's an interesting suggestion. Right. Yep. Thanks. So right. How much time we have? So let's go to the next step. So so the output of the neural nets is the read call per read call, and it's basically probability of how likely this base is methylated. And then because there are many reads that map to one position, and you might have different calls for each of them. And how would you do? How can we predict if this position is methylated? And this is the second part of the project. So the state-of-art method, as I just mentioned before, they use methylation frequency threshold, and it's something around 0.5 to 0.7. And if it's above this threshold, they call this position methylated. Otherwise, it's not. We designed a Bayesian model for doing this task, like the graph we show on the side. So we defined there are 2 states for every call. It's either methylated or it's not. In this case, it's either 1 or 0. And then S is the true state of a position on the genome, right? And then R is the true state of a read. And then X is our observation. So S and R are both hidden. We cannot see. We could only see the X, which is our model's call on each read. And we try to infer what the state of S. And this is so we model, first of all, we model R with a Bernoulli distribution, and these 2 parameters are predefined. Epsilon and gamma are predefined. And then we model the observation with a beta distribution, and there are while R is 0 and X is subjected to a beta distribution that's parameterized by A and B. And these 4, A, B, C, D, these 4 parameters in our case, we estimated using the data. So it's basically telling you if this position, the true state of the position is negative, and what is the distribution of the read that map to this, the read state that map to this particular position. And this bit is telling you that if the model, if the true state of the read is 0 or when it's 1, we are modeling the observation. That is the call from our model. I hope that's clear. Yeah? Okay, going fast. So yeah, so eventually we end up with this equation, and we are trying to compute its likelihood, where if S is where S is negative or S is positive, and we try to compare this to likelihood to see which one is greater. Then we will call this particular position methylated or not. Okay, and to the experiments, we use 3 different data sets and containing 2 types of different DNA methylations. And these 3 data sets are published and publicly available. And they are E. coli data, human data, and plasmid DNA data. So this is on the read base. This is our performance on the read base, and we got the best F score and accuracy and recall benchmarking against 4 different state-of-art methods. So, and then we have these graphs showing the read-based performance, and then these are the position calling performance. So this I want to point out this part that using our Bayesian model, we actually have a quite good performance on different proportion of the methylation reads. So the experiment is that we use 5 different data sets, that it's a mixture of proportion of the methylation read. And our methods performed great in all of them. So even for some very low percentage of methylation reads, we are able to call a quite high accuracy. So, and that is more graph showings on humans and plasmid DNA data. And then we also compare with the state-of-art methylation calling method, which is piece-of-wide sequencing data, and we have a high correlation with it. Right. Right. To summarize that. In this method, we combined both the signals information and the base calling errors to detect DNA methylations. And we propose a novel Bayesian model for position state calling, which results in high correlation to the state-of-art prediction methods. And we show improvements on benchmarking against the state-of-art methods on both read level and position level on 3 different species. And the publication is available on Bioinformatics. And this is a joint work with Pepe. And these are funded by Contra. And thanks to our supervisor, Jens and Nulia, and our colleagues, Abel, Mark, and Simon in Barcelona. Right. Thank you.
So, sorry, I saw the graphical model and all the variables. Can you show again?
Sure.
I mean, all this distribution, the beta and the Bernoulli, and I mean this. I mean, yeah, the gentleman said a couple of minutes ago that neural networks, like probably other method can work. Even HMC. I mean, this is our continuous distribution. I guess with a very low very low amount of code you can use Julia.gl or Turing, and you have HMC. And probably, I mean, I'm not saying that this method works perfectly, but is there another? I mean, do you try before using neural network? You try another type of inference?
Not particularly in these. How to say? Not on this problem setting, I think. So the one I showed before, which is, sorry. Why is it moving now? Okay. The upper method, they are not using neural nets. They're using a simple PCA. And the performance, I would say, not as good as the neural networks. So we didn't start with some simple method in this case, because this protocol of processing raw data to partially to the input to neural nets are already available. So we just take a bit of it and develop our own method. But I think it might be interesting to try some more simple models. Thanks.
Any other question?
Did people ever try, I mean, I just wonder, is there a method out there that just uses the raw signal and then just tries to predict base pair plus methylation as a sort of end-to-end?
Probably there are. Probably are.
So how do they perform compared to that? Is there like or what's the downside of that?
I think, I'm not sure about that actually. Yeah, I need to check the paper. But because we are benchmarking against the state of art, and also because the nanopore sequencing people developed this method called, which I didn't emphasize, but Magaladon is the one they proposed, and it's claimed to be the state of art, and it's a neural network. But they are not open source, so we don't know what kind of architecture they have. So yeah, I suppose neural networks works better in this case. More question?
Okay. So thank you so much one more time.
Thank you.